We all know that reproduction is the only process by which life is maintained on earth, and we have also learned that reproductive processes give rise to new individuals that are similar in structure from that of parents yet different from each other. As a result of different modes of reproduction, some amount of variation is produced whether the organisms are produced asexually or sexually. Each generation provides the next generation with a common basic design and some small changes or variations. These variations get accumulated and are passed on to more and more individuals with each generation. Genetics deals with the study of heredity and variation. The transmission of characters from one generation to next generation is called heredity, while the difference of characters between parents and offspring or between the species is termed as variation. Now we shall learn in detail about the formation of variations during reproduction and how they are passed on to the next generation. In the case of asexual reproduction, single parent is involved and hence the transfer of genetic material is from single parent only. Due to this reason, not much variation is seen in these type of offsprings. Only little changes can be seen because of the inaccuracies happening during DNA copying mechanism. In the case of sexual reproduction, two parents are involved. There is fusion of male and female gametes resulting in the mixing up of different characters. This difference in genes causes maximum number of variations in their offspring and they are unique in nature. This variation which is accumulated in an offspring is passed from one generation to other generation and this maintains genetic continuity of an organism. These variations are noticeable in nature. Depending on the nature of variations, different individuals have different kinds of advantages. The variations are passed on to the next generation through genes. The process by which organisms acquire these characteristics from their parents is called heredity. The process of heredity can be referred to as inheritance, where the information about various characteristics are shared from parents to offspring. These characteristics are known as traits, which is any observable feature present in a living organism. Traits are of two types: inherited traits, which is genetical in nature, and acquired traits, which are developed by an individual during his or her lifetime. Every individual has a unique set of traits. The traits which are transmitted by the parent to its offspring during the process of fertilization are called inherited traits. This inheritance is determined by certain rules of heredity. Inherited traits are coded in our DNA and hence can be passed on to the next generation. Examples are eye color, height, skin color, hair color, etc. Before moving deep into the session, let's familiarize some terms. First and foremost, a gene. It is the basic unit of heredity. It's a segment of DNA that controls a character. They are seen in chromosomes, which are thread-like structures found in cell nuclei. The characters which are observed or transferred from one parent to offspring, or in other terms progeny, is called traits. Traits are of two types: dominant traits and recessive traits. Dominant trait is the visible outlook of the character on an individual, whereas recessive traits are the characters which are hidden or suppressed in an organism. Each individual has a gene responsible for one character. For example, genes responsible for height are represented with alphabetical letters like capital T, small t, where capital T stands for the dominant allele and small t stands for recessive allele. Allele is basically a set of genes which can be seen as pairs or in multiple appearances in chromosomes. They can be of two types. First type is a homozygous allele. where both alleles of a particular gene are the same. For example, capital T and small t small t. Second is heterozygous type, where both the alleles of a particular gene are different. Example, capital T small t, which codes for tallness. Phenotype refers to the physical characteristics of individual. For example, observable height of an individual. Genotype refers to the genetic makeup of an organism. In other words, it describes an organism's complete set of genes. Always, fusion of gametes is expressed as crosses in genetics. There are mainly two types of cross. One is a monohybrid cross, where two individuals with homozygous genotypes, that is capital T into small t, which result in the opposite phenotype for a certain genetic trait. Here, capital T and small t are monohybrid traits here. The genotypic ratio of monohybrid cross is 1:2:1. Dihybrid cross is the cross between two different genes that differ in two observed traits. The classic example for dihybrid cross is Mendel's experiment. The phenotypic ratio of dihybrid cross is 9:3:3:1. Rules for the inheritance of traits. The rules for inheritance of traits in human beings are related to the fact that both mother and father contributes an equal amount of genetic material, that is DNA, to offspring. So an offspring will get two versions of that trait from the two parents. Mendel worked out rules for inheritance of these traits. Gregor Johann Mendel is regarded as the father of genetics. He worked out the first ever scientific experimental study on heredity. Mendel, an Austrian monk, observed variations in the characteristics of garden pea plants, that is Pisum sativum, which he cultivated in his monastery garden. Mendel was curious to find out the results of crossing of pea plants with the variation in traits. Mendel started an experiment blending his knowledge in science and mathematics on pea plants. The main reasons for choosing pea plant for his experiment were: the flowers of pea plant are bisexual. Characteristics are easily visible. The plants are easy to maintain. Pea plants are also easy to cross. The generation time of pea plants is less. And another reason for choosing pea plants was that they were self-pollinating, which enabled them to produce the next generation of plants easily. Mendel used a number of contrasting visible characters of garden peas like round or wrinkled seeds, tall and short plants, white or violet flowers, and so on. The cross in which Mendel showed inheritance of dominant and recessive characters is a monohybrid cross. Here the inheritance of only one character is used. He took pure breeding tall and pure breeding dwarf pea plants and cross-pollinated them to obtain first generation or first filial generation. In the F1 generation, he obtained only tall plants. This meant that only one of the parental traits were seen, not the mixture of two. Now, let's construct a Punnett square. It's a graphical representation of the possible genotypes of an offspring arising from a particular cross. In pea plant, for tallness, the dominant gene is capital T and the recessive gene is small t for dwarfness. As per the Punnett square, all F1 generation progeny has all uppercase T and lowercase t progeny. All of the plants were tall. It was also seen that there were no plants found having medium heights. You can understand that dominant character is exhibited and no dwarf pea plant or short pea plants were obtained in the first generation of progeny. Dominant trait is the trait which is expressed in F1 generation. Here tallness is the dominant trait and shortness is the recessive trait. The plants of F1 generation are then self-pollinated to obtain the F2 generation. Now all of them were not found to be tall. He obtained 75% of tall plants and 25% of dwarf plants. That is, the phenotypic ratio was 3:1. This indicates that in the F1 generation, both tall and dwarf traits were inherited, but tallness expressed itself. Tallness is a dominant trait here and dwarfness is a recessive trait. F2 generation has a genotypic ratio of 1:2:1 of the three types of plants represented by capital T, capital T, capital T small t and small t as shown in the crows. So the conclusion is phenotypic ratio is 3 tall is to 1 dwarf and the genotypic ratio is 1 pure tall is to 2 hybrid tall is to 1 pure dwarf. Dihybrid cross. Mendel also carried out experiments to observe inheritance of two pairs of contrasting characters, which is called dihybrid cross. In the F1 generation, he obtained all round and yellow seeds. It means round and yellow traits of seeds are dominant features while wrinkled and green are recessive. He self-pollinated the plants of F1 generation to obtain F2 generation. He obtained 4 different types of seeds: round yellow, round green, wrinkled yellow and wrinkled green in the ratio of 9:3:3:1. The ratio can be obtained accurately with the help of a Punnett square. How do the traits get expressed? Cellular DNA is the information source for making proteins in the cell. A part of DNA that provides information for one particular protein is called a gene for that protein. For example, the height of a plant depends upon the growth hormone, which in turn is controlled by the gene. If the gene is efficient and more growth hormone is secreted, then the plant will grow tall. If the gene for a particular protein gets altered and less of it is secreted, then the plant will remain short. Both the parents contribute equally to the DNA of the next generation during sexual reproduction. They actually contribute a copy of the same gene. For example, when a tall plant is crossed with a short plant, the gametes will have a single gene either for tallness or for shortness. F1 generation will get one gene for tallness and another for shortness also. How do gametes get a single set of genes from the parents who have 2 copies of genes in them? Each gene set is present not as a single long thread of DNA, but as a separate independent piece, each called a chromosome. Each cell gets 2 copies of the chromosome, one from each parent. Each germ cell or gamete has 1 copy of it because there is reductional division in the sex organs at the time of formation of gametes. When fertilization takes place, the normal number of chromosomes is restored in the progeny, ensuring the stability of DNA of the species. Mendel formulated few principles on the basis of the experiments conducted and concluded it as laws of inheritance. First is the law of dominance, which states that one of the inherited traits will be dominant and the others recessive unless both the factors are recessive. In other words, a dominant gene will express itself over the recessive gene. Second is the law of segregation. It states that during gamete formation, the genes of a particular character separate and enter different gametes. The third law is the law of independent assortment. It is based on dihybrid cross and states that genes for different traits are sorted separately from one another such that the inheritance of one trait is not dependent on the inheritance of other trait.